Greetings to everyone and welcome back once again to this educational channel called Learn Bio with Janet. I'm teacher Janet and I hope that you are fine and coping well. Today we'll be studying Mendel's first law, and this subtopic is from Form 5 Biology KSSM syllabus, Chapter 11 entitled Inheritance. This is the fourth video, part 4, for subtopic 11.1. So in this video, we're going to discuss the brilliant conclusions that Mendel made concerning his experiments that are related to monohybrid inheritance. All right, so let's begin. The learning standards for today's lesson are as follows. Subtopic 11.1 Monohybrid Inheritance. So we have finished studying the first few learning standards, and now we are on to number 4, Construct Monohybrid Cross Diagrams, which we will continue to practice drawing in the questions that will be given to you. And number 5, determine the genotypic and phenotypic ratios of filial generations in a monohybrid cross. Number 6, describe Mendel's first law. And also we will be studying how to draw a Punnett square. Let us take a look at the overview for subtopic 11.1 on monohybrid inheritance. Now in the first video, we have discussed the meaning of the monohybrid cross, Mendel's experiment, the procedure, results, explanation, and we learned to draw genetic diagrams. In the second video, we discussed genetic terms, the meaning of genetic terms. And then we also had a third video where we did some exercises. So in this fourth video, we're going to study the summary of Mendel's monohybrid experiment and what his conclusions are about his experiments. And then we will study the first law that he came up with, which is Mendel's first law, or the law of segregation. Apart from that, we are going to discuss how to draw the Punnett square to determine the genotypes and phenotypes of offsprings. All right, so Punnett square and summary of Mendel's monohybrid experiment and Mendel's first law will be discussed in this video. Apart from that, we will try some exercises and apply our knowledge again to construct the genetic diagrams and answer questions. First of all, we are going to summarize the conclusions from Mendel's monohybrid experiments. What conclusions did he make from his monohybrid experiments on the pea plants? So we can recall back and refer back to Mendel's experiment on the inheritance of height in pea plants, okay, which we discussed from the very first video, all right, on chapter 11. So monohybrid experiment, a monohybrid experiment or monohybrid cross involves the study of the inheritance of just one characteristic each. Okay, for example, characteristic of height. So we are going to use this cross between two heterozygous plants, that is big T, small t cross with big T, small t, as an example to explain the conclusions from Mendel's experiments, right? And this is actually a cross which we discussed in the first video, where the F1 generation plants were self-crossed. But if you have forgotten that, just remember it is a cross between two heterozygous plants. So let's refer back to the schematic diagram again. Now the F1 phenotype, first filial generation phenotype, according to the video, the first video, is that both plants are tall. Or we can just call it parental phenotype. Both plants, the observable characteristic of phenotype is that they are tall. And what is their genotype or genetic composition? So both of them are heterozygous. That means in each plant, each diploid cell has one dominant allele for tall stem, which is represented by big T, and one recessive allele for dwarf stem, represented by small t. Okay, so they are both heterozygous plants, having one dominant and one recessive allele in each of their diploid cells for the characteristic of height. Okay, as seen here, as is pictured here. Now, in meiosis, the homologous chromosomes separate from each other, thus they will carry their alleles, okay, together with them. So the alleles will separate from each other. Big T will separate from small t in the formation of gametes. So each gamete will have only one allele for the characteristic of height. For example, big T is found in this gamete and small t in the other gamete. That means 50% of the gametes from this tall plant have allele big T, and 50% of the gametes from the same tall plant have the allele small t. Similarly for the other parent too, because the other parent has the same genotype as the first parent. Then in fertilization, the gametes will fuse together, right, to form the zygote or the offspring, all right, which is the F2 generation. So F2 genotype or offspring's genotype, right. So first gamete can fuse with first gamete from the other plant. So these two alleles will come together in the zygote. So this zygote here, or the offspring, will have the genotype big T, big T. And then first gamete with second gamete, because fertilization is random, so there are various possibilities for the fusion of gametes. So big T, small t will be found in this zygote. Now this zygote will have the genotype big T, small t. Second gamete with first gamete, the third option. Again, we get the genotype big T, small t for this zygote. And lastly, second gamete can fuse with second gamete here. The genotype of the zygote or offspring will be small t, small t. Then from the genotype will determine the phenotype of the offspring, right? So all those offspring with the dominant allele for tall stem big T will be tall. So there are 3 tall and is to 1 dwarf. Then the genotypic ratio will be 1 big T is to 2 big T small t and is to 1 small t. Phenotypic ratio is 3 tall is to 1 dwarf. So let's use this example of this schematic diagram to explain Mendel's conclusions on his monohybrid experiments. So let's look at the various conclusions that Mendel made and that we can make from his monohybrid experiments. Now all these conclusions have been discussed before, so we are just going through it one final time. All right. So number 1, a characteristic inherited by the next generation from a parent is determined by a genetic factor, which Mendel called heredity factor or inheritance factor. And now we know it as a gene. Okay. Now, so we can take the example of the characteristic of height. So the characteristic of height inherited by the next generation from a parent is determined by a genetic factor or a gene, correct? Okay, so the characteristic of height in pea plants, for example, is determined by the gene for height. Second conclusion, an organism's trait is controlled by a pair of genetic factors called alleles, found in each diploid cell of the organism. Okay, so we look at this example of this tall plant which has the genotype big T, small t. So its trait, which is tall stem, is controlled by a pair of genetic factors, or which we now call alleles, found in each diploid cell of this organism. So it is the interaction between these two alleles that determine the characteristic or the trait of the organism here, which is tall. The plant is tall. All right. So from here we can see that this plant has the genotype big T, small t, and because big T is the dominant allele for tall stem, it will express its trait, whereas, and also it will suppress this the effect of the recessive allele, which is small t. Okay, so it will cause the plant to be tall. So an organism's trait is controlled by the alleles. Thirdly, during formation of gametes, a pair of alleles segregate or separate randomly. Only one allele from the pair is found in a single gamete. Okay, so we are coming to this part. Now, during the formation of gametes, that means during meiosis, a pair of alleles segregate. Segregate means to separate. That means they move apart from each other. So big T will move away from small t, all right, when the homologous chromosomes separate and move to opposite poles. And only one allele from the pair is found in a single gamete. So in a single gamete like this gamete, only one allele is found. In this case, it's big T. And for this gamete, only one allele is found, and that is allele small t is found in this gamete. Not two alleles. We don't have two alleles in one gamete, but only one allele in each gamete. Now, fourthly, during fertilization, a zygote form possesses two alleles, one allele from each parent for a specific characteristic. So now we are at this part here. So during fertilization, okay, fertilization here, according to our schematic diagram, a zygote form, for example, this zygote, possesses two alleles, for example, big T, one allele from each parent, one allele from the male parent, one allele from the female parent, okay, for a specific characteristic. So the zygote will have two alleles in its cell, just like in the diploid cell of its parent. There are two alleles here. Zygote also has two alleles, one allele from each parent. Now same for this one. This zygote, which has the genotype big T, small t. So it received one allele from the male parent, that is big T, and one allele from the female parent, which is small t. So the zygote has two alleles, one allele from each parent for a specific characteristic, for example, for the characteristic of height. Now, the fifth conclusion is that fertilization is random. All right, so random here in BM we say rawak. Perseniawaan berlaku secara rawak, right? As can be seen here, that means that any gamete from the first plant can fuse with any gamete from the second plant here. Okay, any gamete from the first parent can fuse with any gamete from the second parent. That's why we have various possibilities. For example, first gamete fuse with first gamete from the other plant, we'll get this genotype. And then first gamete with second gamete from the other plant. Second gamete with first gamete. And then second gamete with second gamete. Okay, so this shows that fertilization occurs randomly, and there are various possibilities for the genotypes of the zygotes that can be formed. Then an organism with a genotype which is homozygous dominant shows the dominant trait. Now, which genotype is homozygous dominant? By now you should know, right? Yes, it's this one. Big T, big T. So homozygous dominant organism means an organism with two dominant alleles in each of its diploid cells. Okay? So organism with a genotype which is homozygous dominant, for example, this one, big T, shows the dominant trait. Yes, of course, it's tall because it has the dominant allele for tall stem, right? Which will determine that the plant is tall. Secondly, organism with a genotype which is homozygous recessive shows the recessive trait. Now, which genotype is homozygous recessive? Yes, it's this one. That homozygous means having two identical alleles. And in this case, if it's recessive, it means it has two recessive alleles. Okay, so this plant or offspring shows the recessive trait because there is no dominant allele big T to suppress its effect. It only has the recessive allele for dwarf stem. So it will show the recessive trait, which is dwarf stem or dwarf trait. Lastly, an organism with a genotype which is heterozygous, having a combination of one dominant allele and one recessive allele, shows the dominant trait. So it's referring to this organism which has the genotype big T, small t. This is the heterozygous plant or organism with one dominant allele big T and one recessive allele small t. Now it will show the dominant trait. Why is that so? So that is based on the principle of dominance, which we are going to discuss in the next slide. Also, this is also part of Mendel's conclusions. Okay, so it shows the dominant trait because it has the dominant allele big T, right? We're going to discuss the principle of dominance later on, okay, in the up and coming slides. So here are the notes concerning the summary of Mendel's monohybrid experiments and his conclusions that he made, which are all found to be correct based on our knowledge of inheritance in the modern times. So you can copy this now down, and if you are not sure about any one of these statements, please refer back to the previous slide where I've explained each of these conclusions in detail. So what is the principle of dominance which Mendel proposed? In the textbook, it's called the dominance principle, so it's the same thing. So for this principle, okay, which we have actually studied in the previous videos, Mendel explained that in a pea plant with a pair of contrasting traits like tall stem and dwarf stem, one trait is determined by a dominant allele, for example big T, which is the allele for tall stem. Mendel used the term dominant inheritance factor instead of dominant allele, okay? And whereas the other contrasting trait is determined by a recessive allele or recessive inheritance factor, such as small t, which is the allele for dwarf stem. Now, the dominant allele big T, for example big T, suppresses the effect of the recessive allele small t. So, suppress means to menutup or menindas in BM, right? That means it covers the effect of the recessive allele, so the recessive allele cannot express its trait. Hence, the recessive allele is not, the recessive trait is not visible. The recessive trait is not visible, although its allele exists together with the dominant allele in a pea plant. So for the pea plant, which is heterozygous, having one dominant allele, big T, and one recessive allele, small t, only the dominant allele is able to express its trait, and it suppresses the effect of the recessive allele, small t, so the plant is not dwarf, but it is tall, as the dominant allele expresses its trait and suppresses the effect of the recessive allele, small t. So the important thing here is to know the effect of the dominant allele on the recessive allele. Okay, that means this point here, dominant allele suppresses the effect of recessive allele, so that the recessive allele cannot express its trait, and only the dominant allele will express its trait in a heterozygous plant. We have just discussed Mendel's conclusions about monohybrid inheritance. Let us now go on to the first law that he proposed. It's called Mendel's first law, or the law of segregation. Segregation means separation. The law of separation of alleles in the formation of gametes. So let's read the first law. He said that a characteristic of an organism is controlled by a pair of alleles, and only one allele of the allelic pair is inherited in a gamete. Right? So the basis of this law is that the two alleles segregate or separate during meiosis in the formation of gametes. Now, Mendel did not know anything about meiosis because it was not yet discovered in his time. But he was able to propose this law, which is based on meiosis. And let us understand what he is trying to say and memorize the law. All right, let's read Mendel's first law again. A characteristic of an organism is controlled by a pair of alleles. So, taking the example of this organism, this plant, pea plant, which has the genotype big T, small t. So, the height, the characteristic of height in the pea plant is controlled by a pair of alleles, for example, allele big T and small t in each of its diploid cells. And only one allele of the allelic pair is inherited in a gamete. Now, allelic pair means the pair of alleles here, big T and small t. So only one allele out of this pair is inherited in a gamete. okay, will be found in each gamete. This is so because the two alleles will segregate from each other during anaphase 1 of meiosis, just as the homologous chromosomes separate from each other, because the alleles are carried by the chromosomes. Okay, so one allele is carried by the paternal chromosome, one allele is carried by the maternal chromosome in a pair of homologous chromosomes. So during anaphase 1, the paternal chromosome and maternal chromosome will separate from each other and move to opposite poles. So they will carry their alleles with them. So the gametes form will have only one allele, okay, for the related to the characteristic of height instead of two. Okay, so actually we have been practicing this law when we draw the schematic diagram for the inheritance of a characteristic. Okay, and that's it for Mendel's first law. So you have to memorize this particular law, memorize the two sentences here, and understand what it is referring to. It's actually referring to what happens during the process of meiosis in the formation of gametes. Now let's move on to something else. Punnett square, right? What is the Punnett square? Now, Punnett square is just an alternative way to determine the possible genotypes and phenotypes of the offspring of a cross, which is formed through fertilization. Okay, so normally in our genetic diagram, we have learned how to use this, what I call the crisscross method, using lines to show the combination of gametes in the production of the zygotes, right? And we use these red lines, green lines like that to show the combination of first gamete with first gamete from the other parent, then first gamete with second gamete with first gamete, second with second, to show the different combinations of gametes in the production of the zygotes or the offspring. So instead of doing this crisscross method, there's another way where we draw a table to determine the possible genotypes of the offsprings as shown here, and also to determine the phenotypes. Okay, so let's have a look at how the Punnett square is constructed or drawn. Now let's learn how to draw a Punnett square. Let's say you're asked to draw the Punnett square for the cross between two heterozygous plants, as in the previous example. Okay, so a cross between two heterozygous plants, big T, small t, cross with big T, small t. All right, so it's very easy. First of all, we must know that if only one characteristic is studied here, is investigated here, that means the characteristic of height, for example, only one characteristic is involved, then this is monohybrid inheritance, and for monohybrid inheritance, the Punnett square is a 3 by 3 table. Okay, because there's another type of inheritance called dihybrid inheritance, where the Punnett square is slightly bigger. Okay, so remember, draw a table. So take a ruler and pencil, and then you draw a table with 3 columns, 1, 2, 3, and 3 rows, 1, 2, 3. Okay, but the first column here is bigger because you have to write the titles at the top here on the top left-hand corner. This is the top left-hand corner. Okay, so first, use your ruler to draw a diagonal line across this box here. So I always say use a ruler because some students don't have a ruler, or they just simply use their hand and draw. So you cannot, now you have to use a ruler to draw the table neatly. Okay, right, then draw a diagonal line across here, and on top here you write male gametes. That means we are going to determine the alleles in the male gametes. Alright, so this symbol here, as is shown in your textbook, this is the symbol for male, and this is the symbol for female. So this other box here, this other triangle here, space here, is for you to write female gametes, and the alleles in the female gametes will be shown below here. Okay, so let's say this first plant, big T, small t, this plant with the genotype big T small t. Okay, it's the male plant, all right, or the plant that contributes the pollen grains or the male gametes. So what are the male gametes for this plant? Okay, according to Mendel's first law, each gamete will have only one of the alleles shown here. So male gamete number 1 will have the allele big T, and male gamete number 2 will have the allele small t. Okay, so you just write the alleles here, right? And for the female gametes, is also from the heterozygous plant. So for the first type of female gamete, for the first type of female gamete, the allele is big T. Okay, and for the second type of female gamete, we have the allele small t in it. All right. So as I said, it's not just one gamete each here, but 50% of the gametes will have allele big T, 50% of the gametes will have allele small t. Okay, similarly for the male gametes here. All right, then after that, you have to fill in the boxes here. So these boxes here, the boxes on the bottom right, which are all these boxes here, show the different allelic combinations or combination of alleles that can be found in the zygote and or the offsprings that are produced. So easy, very easy. Just take this gamete with the allele big T. So this allele big T comes down here, and this allele big T comes down here at the intersection here. Okay, so the first genotype. Okay, now all these 4 boxes here show the genotype of the offsprings or the zygote that are produced, the zygotes that are produced. Okay, so first genotype here of the zygote or offspring will have the genotype. I mean, the offspring will have the genotype big T, big T. Now, how about the genotype for this second offspring? Just write in this big T and small t alleles. Okay, and then for the third one, so you can imagine, just draw a line down here, So these two will come into this space here, okay, where they intersect, where there's an intersection of lines. So big T, small t. And the fourth one, small t, small t. Okay, so these are the 4 genotypes of the offsprings that are produced. Okay, and from these genotypes, we can determine the phenotypes, right? For example, all those zygotes with the alleles big T will be tall, and the homozygous recessive organism here, or offspring here, will be dwarf, because it doesn't have any allele big T, so it will express, the small t allele will express its trait, which is dwarf trait. Then from this result here, okay, from here we determine the genotype and phenotype. We can determine the ratios and probabilities of the genotypes and the ratios and probabilities of the phenotypes. For example, what's the ratio of the genotypes here? So group all the genotypes together, the different genotypes, and we will get the genotypic ratio of 1 big T is to 2 big T small t, 2 of them here, and 1 small t. Okay, so this result is the same as when you draw the whole schematic diagram using the crisscross method for fertilization, as we have seen in the previous slide. Okay, when you draw the schematic diagram. But here we're using Punnett Square to determine the genotypic ratio. And for the phenotypic ratio, it's 3 tall to 1 dwarf. Okay, so, and we can even determine the probability of getting a genotype. For example, probability of getting this genotype in the zygote. Probability of getting an offspring with a genotype big T. What's the probability? So if you express it as a fraction, it will be 1 out of 1, 2, 3, 4. 1 out of 4. The probability of getting or producing a zygote or offspring with the genotype big T, or homozygous dominant offspring. Okay, so this is what the Punnett square can do, and this is how you draw a Punnett square. Remember, for monohybrid inheritance, the Punnett square is a 3 by 3 table. And also remember the titles at the top here. These are the alleles in the male gametes. and the alleles in the female gametes are written here. Okay, a question here. When can we use the Punnett square in our drawing instead of the crisscross method? So my tip or comment here is that normally we do not draw the Punnett square unless we are requested to do so in a structured or essay question. So normally for schematic diagrams where we have to draw the genetic diagrams like this, we usually use the crisscross method as shown here to determine the genotypes and then the phenotypes of the offspring. Okay, so all these lines we call the crisscross method is to show us the fusion of gametes from the two parents, right? Okay, so normally we'll use this way of determining the genotypes and phenotypes of the offspring. So this is the original way that we have studied. And we only use the, usually we only use the Punnett square when we are requested to do so in the structured or essay questions. Let us now apply our knowledge to this interesting question. In pea plants, the allele for round seed, big R, is dominant to the allele for wrinkled seed, small r. Based on the key above, draw a Punnett square to determine the offspring's genotypes when a male parent with genotype big R, small r is crossed with a female parent with genotype small r. Predict the genotypic and phenotypic ratios of the offspring. Okay, and the key above is given. Big R represents allele for round seed, is the dominant allele. Small r represents allele for wrinkled seed, which is recessive. Right, so the first thing you need to do is to highlight your important information. So allele for round seed, big R, dominant to allele for wrinkled seed, small r. That's the first part. Then you're supposed to draw a Punnett square, right, to determine the offspring's genotypes. And the cross is between a male parent with genotype big R, small r, or sometimes they may say a heterozygous male parent, because this type of genotype is called heterozygous. And cross with female parent with genotype small r, or a homozygous recessive female parent, or just a female parent with the recessive trait. Okay, which is wrinkled seed. Now, then you're asked to, after drawing the Punnett square, you must predict the genotypic and phenotypic ratios of the offspring. Okay, and also based on the key. So the key is based on the information here. Okay, so the key has been given. Now, first of all, notice that you are supposed to draw a Punnett square, not the whole schematic diagram. All right, so you don't need to draw the whole schematic diagram here. Just draw the Punnett square. And then there are two types of Punnett square, but for the one that involves only one characteristic or monohybrid inheritance, the Punnett square has dimensions 3 columns and 3 rows. 3 columns by 3 rows. Okay, so here only one characteristic is studied, and that is the type of seed. Let's look below here. So for pea plants, there are 2 types of seeds. All right, the first type is smooth and round. Second type is a bit misshapen and wrinkled on the outside like this, right? So round seed and wrinkled seed. So round seed is dominant. The trait is dominant compared to wrinkled seed. Round seed trait is dominant compared to wrinkled seed. All right, so now we have all the information here. You have to draw your Punnett square. Okay, so for monohybrid inheritance, when only one characteristic like the shape of the seed is involved, and there are 2 contrasting traits, round seed and wrinkled seed. So for this type of inheritance, the Punnett square is 3 by 3. So you can start drawing your Punnett square and then pause the video, draw it, and then after that, just compare your answer with the answer here to see whether you have understood what you have learned and be able to apply. Okay, you can pause the video. So I will go on to the answer in a few seconds time. All right, let's look at the answer. So your Punnett square has 3 columns and 3 rows, right? The first column is broader because you have to write the words male gametes and female gametes. So this top top left hand box here, you have to draw a slanting line down like this, like this, and then write male gametes above, female gametes below. Okay, so these two boxes are for the male gametes. Now look at the male parent. Male parent has genotype big R, small r. So according to Mendel's first law, during formation of gametes, the alleles will separate, okay, and only one allele is found in each gamete. So the first gamete has big R allele, the allele for round seed, and the second gamete, second type of gamete, has the allele small r or allele for wrinkled seed, okay, according to the key here. For female parent with genotype small r, the two small r's will separate during meiosis. So the first gamete has the allele small r, and the second one also has the same allele small r. All right. Now, what are the results of the cross? That means the genotypes of the offspring. So big R, the first male gamete has allele big R, and the first female gamete allele big R. So here, where they intersect here, what is your answer? So the first offspring has genotype big R, small r, right? And then the second one, genotype small r. Now the third one below here, this one and this one. Again, big R, small r. And this last one, small r. Okay? Right, then these are the four genotypes of the offspring. So what is the genotypic ratio? So gather together all those with the same genotypes, like big R, small r, the heterozygous offspring. How many are there? 1, 2. Okay, and for the homozygous recessive offspring, small r, with genotype small r, there are 2. So 2, 2. So 2 big R, small r is to 2 small r. Simplified, you get 1:1. Okay, so this is your answer for the genotypic ratio. Now, phenotypic ratio means the ratio of the observable characteristic or trait, the trait. So determine first of all these two big R, small r, the heterozygous plant. What type of seed did it have? Okay, what type of seed? So since there's a big R allele here, it must express the dominant trait, and dominant trait is round seed. So these two plants had round seeds, okay, and the other two, small r, they had wrinkled seeds, because here only the allele for wrinkled seed is present, no allele for round seed. So this plant had wrinkled seeds, okay, these two plants. So phenotypic ratio is 2 round seed here to 2 wrinkled seed. And if you simplify it, it's 1:1. 1 round seed is to 1 wrinkled seed. Okay, so that's the answer. So for the final section here, I will leave you with a question: the inheritance of flower color in pea plants. And you can try this question as an exercise. So in the next video, I will be giving the explanation for this exercise, and also give you some further objective questions to try, okay, with explanations for the answers. Now, question 1. A biologist carried out an experiment to study inheritance of flower color in pea plants. He first carried out a cross between a purebred plant with purple flowers and a purebred plant with white flowers. All the offsprings in the F1 generation produced purple flowers. Okay, that's for experiment 1. And then in experiment 2, he self-crossed the plants of the F1 generation to obtain the F2 generation, self-crossed. Using big B and small b to represent the alleles for flower color, draw 2 schematic diagrams to show the results of experiment 1 and also experiment 2 separately. Draw them separately. And then explain how the F1 generation with purple flowers is produced. So here you need to use words to explain, not just drawing a schematic diagram is not enough. Okay, so the first thing you need to do is to highlight the various points. Now in the first experiment, biologists carried out a cross between purebred plant, or purebred, a plant that is purebred with purple flowers, and then a purebred or purebred plant with white flowers. Okay, white and purple, purebred. all the offsprings in F1 generation produce purple flowers. Okay, that gives you a hint which color of flower is dominant, right? Next, he self-crossed the plants of the F1 generation to obtain the F2 generation. Okay, before we go on further, can you tell me which color of flower is the dominant trait? Yes, it is the purple flower, because all the offsprings in the F1 generation produced only purple flowers, although one of their parent had white flowers. Okay, so this is the key to note, that purple flower color is the dominant trait. All right, or the allele for purple flower is dominant to the allele for white flower. Okay, so the two contrasting traits are purple and white flowers. Next, using B, big B, small b, represent the alleles for flower color, draw the two schematic diagrams, okay, to show results of experiment 1 and 2. So you should have two parts here. All right, now then we can construct our key now. So in our key, big B will represent the what? Big B doesn't represent purple flower. It represents what? Allele for purple flowers. Okay, let's write down. And that's the dominant one. Okay, this is the purple flower and this is the white flower. So in the key, so I'll start you off, and then later in the next video, you can check your answer, right? So in the key, let big B be the dominant allele for what color? Purple flower trait, okay? Or the purple trait. And small b is the recessive allele for the white flower trait. Okay? Then with this key, you should be able to draw your diagram very well. Okay? Just refer to the first video which we discussed about the inheritance of height. Okay, that Mendel, the experiment that Mendel carried out. Now, explain how the F1 generation with purple flowers is produced. So you need to explain in words. So just a hint, you have to talk about meiosis and fertilization, and explain how in the end you get F1 generation which produce purple flowers only. Okay, now you can also read up from your textbook, page 234 and 235 in the textbook to get some idea how to draw a diagram. Or you can start drawing your diagram first and then check with that. So I'll be giving you the answers in the next video. All right. So that's all for this lesson. Thanks for watching, and we will discuss more about inheritance and genetic questions in the next video. Please share, like, and subscribe. And goodbye. Take care.